I guess we can get started. I'm not sure if um Mike will be joining or not. So today's lecture will be um talking about the single cell RNA-seq workshop. So basically just run through a quick kind of quality control that we brought up last week. Like we need to make sure there's like good quality enough of cells, and then every cell have enough genes, and then we will then rely on like the dimension reduction normalizations and then finding out the clusters in our single cell RNA-seq data. So before get started into the lab, anyone has any questions um about the previous lecture stuff? Okay, if not, we can get started. Yeah, I already post this link. Oh shit. I already post yeah, I already post this link here on on Brightspace, so you'll be able to like access this link. So um yeah, so just like as we're running like pandas or Seaborn, every time we always need to make sure this library is installed in our Jupyter notebook. And then if you remember Jupyter notebook, Google Colab, like this for Google Colab will just erase everything. So that's why if you want, you can also open up the Jupyter notebook on your like local like MacBook or Windows. So you can just install the library package once and then you don't need to every reinstall this every time. And yeah, so for the library to process single cell RNA-seq data in Python, we will be using Scanpy. This tutorial, I think I also post this link onto the Brightspace. So if you can see it, you can just search Scanpy or Scanpy documentations, and then basically they will have a lot of basics telling you like what's our a standard single cell RNA-seq. We call pre-processing basically just quality control, filter out low quality cells, make sure all the cells and genes are good quality enough, and then they will do clustering. So yeah, the first step we need to first install this Scanpy, and then this is the Scanpy's installation command, like pip install. If you want, you can also look at here installations, the pip part. So what I did, I just copy the code here and then run it, and then paste it to the Jupyter notebook, and then I just execute it, and then they will run it for me. So if we are, yeah, so after we installed a library, what's our next step? We need we need to like import the library. So in this case, we'll be importing like the scanp as sc. So if there's any like command that using like sc, that basically is that we're using the functions from the scanp tools. And then like these end data as AD is just like a pre-installed software for us to like call or do any like multi-layer data set. So what that means, I will show this in the in downstream like other code block. And then this one just for because I'm downloading a kind of a demonstrating data set from the cloud. So this is just a library for me to download the data. So you can also ignore this. I don't think you will be using this. And yeah. And then like a lot of times Scanpy like you can first set all the figures that you're going to plot for their DPI. So for example, I want to set all the styles the same. So maybe I want DPI to be 300 and then face color to make every of the colors to be in the white format. So this is also one of the kind of a background command. So when you're running single cell RNA-seq, these and these like you can just click it through, don't really need to modify. And then right now we're going to like download a demonstrated single cell RNA-seq dataset from a website. So this is just a quick reference that where which the study are using. So oops. It's not yeah, but I think it's fine. Wait, let's see if I can get the data sets out. Yeah. So this is the data sets here. So it is a kind of data sets for like single cell RNA-seq data, and I think it is for immune cells. It's a little bit, hmm, yeah, but I think it's fine. It's just a demonstrated data sets, and yeah. So like this is just for like the lecture, so I would just like click it through, and I think the most important command here is this one, the sc.read 10x h5. So this is a command that we will be running to read a single cell RNA-seq dataset. So single cell RNA-seq dataset has a lot of different file names. The .h5 is one of the file name. And then if you look at Geo, a lot of the times, oops, file. If you look at Geo, a lot of the times they will have their own single cell RNA-seq. Like, let me pull out the lecture slides. Yeah. So like if you search this here, they will also have their own single cell RNA-seq metrics. And then you can see at like they end with barcode, features, and metrics. So if there's like three files like that, the barcode just the cell barcode, and the features are actually the genes, like the gene features, and then the metric just the raw like cell count metrics, and then this is like the larger size. So if you want to like execute publicly available single cell RNA-seq dataset, or for example, I send my samples out for to do like 10x like genome like single cell RNA sequences, they will also turn me over these three files. And then what I will do, I will just like download them from public available datasets. And then I will demonstrate that in a few in like down in later. And then they will have another command, or maybe just let me scroll all the way down. Like there will be another command called sc.read 10x metrics. So what does that mean? That just means it is we are running the read 10x metric functions to read a standard single cell RNA-seq metric file, which I will maybe let me just show you here. Like I store it here, and then yeah, I basically will store the barcodes, the feature, the metric file under this folder, this filter metric folder, and then I will just directly point out my path to this folder, and then I can just run it, and uh, they will they will be able to like read the single cell RNA-seq metrics. Let me just show you here. So what did I do here? I've used the Scanpy function to read my 10x genomic metrics, and then I save this into the a data one, like a data for like a metrics variables. So it's reading it now. Okay. So now if I want, I already load, oops, I already load my n data one. So it's already loaded here. So what is N data? I think I brought this up before. Like N data is a multi-layer of data sets. So the adata.x, which is the raw gene count metrics. And then they also have like different layers, like the variable layer, which is the gene information, and then the OBS, which is the cell informations. I think these are the three, like the blue color, the yellow color, the green color are the three most commonly used like N data like layers. Yeah. So let me just click it through. So yeah. So right now I'm downloading other sets of single cell RNA-seq metrics, and then I save them into the N data, like N data variables. So now I have loaded like two separate single cell RNA-seq data sets. Okay, so as I mentioned before, the adata.x, which is a layer that is the main table of cells and genes. So what does that mean? It's just like it's like pandas where we are calling like columns. But slightly different thing is you need to first put like adata.x. Yeah, so this is a and then yeah, maybe like adata.ops. You can see this is a sales information. So you can see in this adata.obs, which is a sheet of metadata about like rows. So each rows here are a cell in our single cell RNA-seq data set. So anyone, maybe, yeah, you don't need answers. So how can we know how many cells we have sequenced in our single cell RNA-seq? That's mean we have like 17,000 cells are sequenced. So what does that mean? If you still remember for single cell RNA-seq data sets, we are flowing every cells through this inside a droplet, and then every cells will be hybridized with a unique like barcode. So that just means in this A data, like this study, we like sequence 17K cells, or after the readout, we identified there are 17K cells. And then if you're interested in looking at the adata.var, the variable, the .var like layer, you can also type adata.var. So for this, it basically is like each gene's informations here. And then I think adata.layers, just like sometimes when you normalize your single cell RNA-seq data set, um, no, layers. I think is it count? Huh, wait. Oh, so I haven't have any layers yet. Okay. So I think later on they will generate this here. So I think this is the single cell RNA-seq like multiple layers of your data sets. And it's just like I would say it's like three to like four, five data frames. And then a lot of the time when we're doing like pre-processing, we'll be like adding informations on the .ops layer because ops are like per cells informations. So what does that mean? I think I will just show you later. So this is an introduction of the basic single cell RNA-seq data. How does it look like? So after we load the single cell RNA-seq data sets, we will be doing some we call pre-processing. What does that mean, or what's the high-level concept of that? Basically, we just wanted to make sure the cells and the genes are quality enough for us to normalize, and then for us to do dimension reductions, and then for us to really identify like the different cell clusters in our transcriptomic profile in our single cell RNA-seq data sets. So they will have like three functions in single cell RNAs in Scanpy. If it's sc.pp, that is a function for pre-processing. And then the pre-processing, as I said, just for um make sure the cells are good quality enough. And then if there's sc.tl, that basically just a toolkit in Scanpy to run like downstream analysis. So like the UMAP functions, I will be using the sc.tl functions. And then if sc.pl, that just means it is a plotting function for Scanpy. So we can also like visualize the UMAP. I will show this in downstream. So if you see sc.pp, that just means it is a pre-processing part. sc.tl is a toolkit part for Scanpy. And then sc.pl, which is the plotting functions. Yeah. Any questions? Yeah. So right now, I'm just doing my pre-processing quality control. So like this three code block here, basically I'm wanting to look at if there's like mitochondria genes, if there's like ribosomal genes, if there's like hemoglobin genes, because I think these genes will be showed inside our um yeah, like these genes are not really reflecting our transcriptomic profile. The MT is mitochondria genes. So basically, I'm like what the code here, they just wanted to identify any genes that start with like MT, that just means it is a mitochondria genes. For yeah. So how we identify them is like through the alignment step from the raw FASTQ file, and then they will just like identify those mitochondria genes for us. So yeah. So now, if I execute this code, they will just identify if every genes here, if any genes on this adata.variable layers, if they are mitochondria, if they are ribosomal genes, if they are like hemoglobin genes. So Yeah, so you just click this, execute this cell, and then if you look at the variable, the AData.variable layer about genes information, they will just shows you um if they are um like these three genes. And it's like pandas. If you want to code a sub column of uh like a data's layer, like a data variable, you also use a square bracket. So I think it's a little bit similar to pandas, but you just need to like specify which layer of your end data you are calling. So I hope it's not too like complicated on your end. And then since it's true or false, maybe you can run some like executed like value like counts like basic pandas. I think it would work. So what does that mean? That just means there's 13 genes that are mitochondria genes, like in this case. So a lot of functions here are all like kind of like you can use a basic like we learned from the pandas workshop to be using in single-cell RNA-seq data sets, which is pretty nice. Yeah. So then, in this case, I'm after I identify those mitochondria or like ribosome or hemoglobin genes, I will run this code. So basically, they will just calculate like what is um the number of the percentage or the numbers of like mitochondria. So you can see here, you can now inspect like how many genes are in your count matrix and then the percentage of like mitochondria genes. Yeah, this is basically just one of the kind of the pre-processing steps. So if you look at like um they will if you look at adata.ops, you can see we keep adding some like logs transform like informations here. And I think I tend to just like ignore these because these are just like some quality control stuff. Um So yeah, I think the most important thing is like some of the like mitochondria, the percentage of mitochondria counts per cell, they will just calculate it for us. And then percentage of like ribosomal genes in every cell. Because AData.ops layers are the information for the cell's information. So basically what this code here they are doing, they basically just calculate for each cells what are the percentage of like mitochondria, ribosome, and the hemoglobin, and then we can decide if we wanted to like filter them out. So in here, like I would say the most important, like yeah. So now I'm going to do. Oh, it looks. Oh, oh, I think because the DPI here. Let me just set it back. So yeah, so like right now, I'm running the plotting functions in Scanpy. So Scanpy can directly just basically run the violin plot. And then the functions that we're using Scanpy to run is like the sc.pl functions. And then I'm using the dot violin like functions in this plotting toolkit of Scanpy. So basically, I'm just telling Scanpy what is my like single cell RNA-seq matrix, and then people just tend to name it like A data. So yeah, so I just specify A data and then tell them what is the column that I want to plot the violin plot. So I can also like for example plot um like percentage of ribosomes for example, and then I can remove this. So in this case, they will just shows you a violin plot of the percentage of like ribosome. So I would say like ribosome is not that big of a deal, but then if you look at like mitochondria, because like people think if there's like too many mitochondria, that just means the cells are dying. But then if you're not looking at the cells that are like dying or like unhealthy or like getting stressed, I would say maybe we should consider to remove the mitochondria. So this is like the Violin plot for all the cells. So the cells, all the 17K cells. So I showed this here. So after that, I might decided to like filter out the mitochondria that is like over like 20% or like 10%, for example. Yeah. And then you can also plot like scatter plot using the sc.pl.scatter. So then they will plot the scatter plot. And then you basically just tell them your what is your X on the scatter, what is your Y on the scatter, and then what is your color of your scatter plot, for example. So in this case, I just wanted to visualize what's how many cells I sequence and then how many genes per cells that I sequence. Is it yeah. And then I visualize the color for the percentage of mitochondria like counts inside every cell. So this is what I did. I hope it's not, yeah, any questions. Because, yeah, I think when we're doing single cell, we can decide how many cells we want to sequence and also how the sequencing depth per cell. So sequencing depth, I think we brought up in the book RNA-seq workshop. Sequencing depth basically are just we are telling how many reads we want to sequence. So in single cell RNA-seq, we can decide like just how many cells to sequence, how many cells to flow through the droplet, and then how many like RNAs, how many reads per cells that we will sequence there, and then get a total read count. So this is just like a quick quality control to visualize how's like your cells like sequencing depth. So maybe there's some cells have like yeah. So yeah.
Um,
So
I
any
just
question?
had a question about the previous plot.
Yeah.
Is it also a violin and scatter plot? Like, what do all the points on the plot suggest? The one
Oh,
above this.
this one, right?
No, no, the one above that.
This one.
Yeah, this one. No,
Oh,
I mean,
this one.
this violin plot. Yeah.
Yeah, so this is a violin plot. So just like when we're using Seaborn, so violin plot basically we tell violin like on the y-axis of violin plot, it is just display the values like in your data frame in your column. So it's like the I'm plotting a violin plot. I'm plotting a violin plot to display the distributions of the percentage counts of ribosomes and mitochondria in every cell. So it will be 17K cells here. And then every dot here just reflects what's the percentage of ribosomal count. And then how we get this number of quality control count, basically we just run this code here. And then we need to first do the pre-processing to get the additional information from to get the additional informations of the percent of mitochondria, ribosome, and hemoglobins, and then we can plot the violin. Because if you still remember, the original N data, the OBS, the ops layer, they only have one like sample column. So we need to first execute the pre-processing, and then they will add these informations on the .ops layer, and then we can plot violin plots over every columns here. And why violin plot? We just wanted to kind of visualize how's the distributions of like the variables we're interested in. So in this case, I'm just interested in how's the distribution of how's the cells look like that has how's the cells look like for their percentage of mitochondria. So if you look at the plots on the right side, that just means majority of the cells only have like less than 20% of mitochondria, which that just means it's like because you see the Violin plots here reflects the distribution. So there's like a really large clusters of cells have like less than 10% of mitochondria. So this plotting just make us decide, for example, our quality control threshold, like how high should I use to like how how high should my threshold for mitochondria? Because if you remember, like we wanted to like ensure we don't keep too much mitochondria genes. Like this is the most important thing. So yeah, so you can see here, I want to feel like the upper bound is like at the highest percentage of mitochondria is reasonable, is like 20%. So maybe if I set 20%, so I will remove all the dots, all the cells that is above here, above the 20% on the y-axis threshold. So this is how I want to plot the violin plot for, because I want to use this to have a sense of how many cells I'm like removing. Or you can, yeah, I think this is the easiest way to do. So just like we can have a sense how many cells, if we want to set a mitochondria threshold to 20%, so maybe this can do that. Yeah, we can do that. And then for ribosomal count, I think they can tell note the ribosomal count, but I think in general we don't really filter it out based on ribosomal count because yeah, you can see the distribution are relatively like homogeneous around like all the cells. Yeah. So, but yeah, mitochondria, because on biology, increasing mitochondria cells, that just means we have more like stress or the cells are dying, just like here. Indicating dying, stress, or like rupture cells for RNA leakage. So we tend to we need to filter out cells that showed higher in mitochondria count. That just means the cells are not good quality. The cells are stressed. So that's why we need to plot. Yeah. So that's why I do the plotting first. So in this case, we also just like basically just display how, yeah, it's basically just like other ways to show. So the color here is the most important thing. The color here just like telling you what is the cells that has higher mitochondria count and then have a sense of how many genes are in those cells and then what's the counts, the total gene count in that cells. Total count. How many cells? Yeah, total count. Just this one. I think this is number of genes identified, and then the total counts just how many like how many reads are there and how many genes are identified. I think this is what the best way to say. So these are I'm plotting these two functions. So if you plot here, you can look at the cells that has higher in mitochondria, which is has lighter color. They tend to have like lower genes and then lower total like RNA-seq reads identified there. So we can think that these cells have like lower qualities. So I will try to filter them out based on the percentage of mitochondria count. So yeah. So after we visualize that, kind of have a sense, maybe I wanted to filter out mitochondria percentage less than 10%. So I will just remember this threshold. And then I can start using the pre-processing function to filter out the cells. So in this case, what does this two code here mean? I'm filtering out the cells that has genes less than 100. So you can like keep increasing the threshold like to 200, 300. Just like, but just keep in mind, if you increase the minimal genes, you will filter out more cells. And then I can also do the filter gene function to filter out like I want to filter out super rare genes that only exist that need to exist in at least three cells. So I don't. Oh, you. So in this case, you can also like increase the like the number of genes here. But just keep in mind, if you increase the number of genes, you will also remove like you will have less genes that filter. So what does that mean? Um, if you still remember, the adata.ops of are the layer for per cells information. So right now it's I have 17K cells. And then adata.var layer are the information for the genes. So I have 36K genes. So if I run this code to filter out the cells and genes, we would expect the number of genes and number of cells to like decrease. So you can see I filtered out like 13K low quality genes in my single cell RNA-seq matrix. And then for my adata.ops, I filtered out maybe only 100 cells. So you can keep increasing this threshold. And I would say, like to me, I tend to just start with lower threshold and then click through the pre-processing method, and then um, see if it makes sense. If it makes sense, I don't think we need to like increase the threshold. But this is kind of the minimum, like kind of the minimum people would request. Or I think this threshold people would like to do like 100 or 200. Yeah. This is one of the standard way. But then if your data set still looks weird in downstream, like if you're interpreting them, I think this is the time you can increase the threshold. So after I filtered out low quality cells and genes, one thing that is also important is I want to filter out the cells that has too many mitochondria percentage, too like big portion of mitochondria percentage. So I set the threshold for 10. It's I think the code looks a little bit similar to pandas, but yeah, it's like more straightforward, basically just telling your single cell RNAs, telling just like write the code here, which column you're using in the adata.ops layer, and then what is your value set. It's like less than 10. So in this case, if I just run it here, if we keep visualizing how many cells um are in my adata.ops now, it's we filtered out like we the cell counts drops from um 17K to like 11K. So then yeah, we just remove low quality cells. So there's always some sort of like trade-off if you like filter them. So I can replot the scatter plot for the over total count and the number of genes per count per cells, and then I keep plotting the same one, and you can see the heat map color now the maximum is only 10. So then yeah, I just decided to go for this single cell RNA-seq metrics. Is it too complicated? I think for you, just you don't need to really change everything. Because when I first learned single cell RNA-seq, I also start with using people's like notebooks and then just click it through. But yeah, and then I think if you click through like twice or like third time or like 10 times, you'll be more familiar now. So I think we brought up the concept of doublet. So what is doublet? Doublet basically is when a droplet are incorporated with like two cells, then that's not single cell RNA-seq. So we wanted to run the sc.pp.scroblet functions. So this will help us identify. They will just use an algorithm to predict if this cell is a doublet or not. So basically, they just look at the number of UMIs, and then if it's like higher, they will just like predict. And then you can set the threshold for doublet. So they will just like calculate the percentage of like doublet and then or like the likelihood, the threshold. And then if you set here, so what does that mean? They will have a kind of a column with a predicted like values, and then anything that is higher than 0.25, they will just predict them as a doublet. So just like kind of like a p-value threshold, but then like this threshold is like likelihood that this cell is a doublet. And then I just set a threshold to 0.25. I think this is also a common threshold like people will use. So if I run sc.pp.scroblet, they will just start just like pre-processing them. And then this is one of the function that takes longer time. So if I finish running this, we will expect they keep adding more columns, more informations to my .ops .ops layer of the single cell RNA-seq data sets. Yeah, need yeah. Before, yeah, maybe waiting for anyone have any questions? So I would say single cell RNA-seq is pretty like it's just similar to pandas. This is what I feel. So that's why I feel like it's a good thing to like brought this up in like the data science class. Um, so yeah, it's just like we learn in pandas to call a column in column of a df. We use a square bracket and then inside a quote we put the column name. It's also the same if you want to call any columns in any layer of single cell RNA-seq. We just use adata.obs and then use a square bracket and then the column name. And yeah, and then just keep in mind the adata.obs are the information per cell. So like Dr. Louis might be interested in the percentage of changes in certain cells. So later on, we can try to collab. Like if we came up with like the cell name, because right now every column, every rows here are per cell. So after we do the pre-processing clustering, we'll be able to add like the cell like name tag to every column here, and then we can also like just take that, just count like different cell types. Yeah. Hmm. Okay, now they predicted. So we would expect they will add some additional information about the scrublet information. So you can look at all the way on the right side. They will have a predicted doublet score. And then I set the threshold for 0.25. So if any predicted doublet score are higher than 0.25, they will be marked as a doublet. So if I want to look at how many doublet is here, I can just call the predicted doublet and then keep using dot value counts. So they just predict that in my single cell RNA-seq, there's like 264 cells that we predict them to be a doublet. Oh, you can also either make, you can either also plot. I think I just wanted to show you. You can also plot like plots using Seaborn, like the dot distribution plot functions, and then in here, basically just tell them one of your continuous variable, and then you will be able to like plot something. Plot with Seaborn. Sorry. So in this case, I'm wanting to like visualize what's the distribution of doublet score in my single cell RNA-seq data sets. So I just use the scborn.displot. And I can also visualize them using Scanpy's like plotting functions. Scanpy.violin. So inside the first argument is your n data, single cell RNA-seq data variables, and then the column you wanted to plot doublet score. So we can see how does it look. So it's just like different ways to plot. So if I filtered out based on like 0.25, maybe they will be like I will cut off like 264 cells that is on the right side here. Wait, am I able? Yeah. I will if I later filter them based on wait, let's see. Yeah. Oh, so I can just oh yeah. So if I want to filter out those those cells that doublet scores lower than 0.25, that I think I will be removing the cells here. And then this is 264. So yeah. Oh, wait. Yeah. So yeah. So this is how I identify the doublet. So it's also just running a one line script and then they can process everything for me. So yeah, so basically what this code here, I'm just wanting to like filter out those cells that is like this is like one of the ways you can filter, or you can just use like this column equals true. You can also filter out the doublet. So but I just this is like the code from the um the tutorials. So in this case, we just execute this code And then this code will help us remove the doublet, which is if it's true, they will remove them for us. So if I look at how many cells now, it also like just remove like 264 of the cells that were predicted with doublet. So after we make sure, so these are called like just like pre-processing to filter out low quality cells, low quality genes, filter out doublet. Then we're gonna like normalize our data sets. Yeah, so the first step here, I'm just like state like I wanted to like create a raw counts layer, a raw count in my single cell RNA-seq because after I start running the normalization, these like counts will be kind of removed. So that's why I want to save this. So I think you don't need to worry, just like execute this code. Basically this is just like I'm saving a raw counts layer and then I'm going to do downstream like normalizations. And why we need to normalizations? Because for single cell RNA-seq, we always need to account for gene length plus sequencing depth. And then sequence in single cell, like every cell will have different sequencing depth. So that's why we need to normalize. And then it's also really, really simple to run. You just use sc.pp.normalize. So then they will start normalizing them for us. And then I would like people tend to like log transform the normalized data sets here. So um yeah, so I just execute them and then it's like did pretty oh wait, a data dot I think. Yeah. So I think this is a raw count we saved. Did I save this? Yeah, but this is like really yeah, really metrics. I don't think we can really visualize them, but yeah, but anyway, I think right now after we run this code, single cell RNA-seq will help us normalize the read counts, and then so it's like really really simple. Like we don't need to just like we don't need to do like the bulk RNA-seq workshop. They will normalize them for us, and then um, important thing for single cell RNA-seq, they will use the clusters with like the top highly variable genes. So what does that mean? They will look at all of your genes and then they will find out top, you can set up the threshold. And then they for in my case, I wanted to set top three, like 22,000 genes that is high variables across all the cells. And then I want to label them as highly variable genes. So basically, when we're doing like PCA and UMAP, they will be using the highly variable genes to do that because they don't want to take all the 20K or 30K cells to do UMAP and PCA. So that's why we will tend to just highlight the highly variable gene. So this is also a kind of a place where you can increase or decrease the threshold. Because if you increase the threshold, you will include more genes to do like PCA, UMAP, and identify the clusters. So I think, yeah, the tutorial just said 2000, so that's why I just picked 2000. So like when to increase or decrease, I think when I wanted to like make sure like if it's the best cluster that they identify, that is when I will use. So if I execute this code here, they will add additional information on the .var, which is a gene informations like layer. So they will just tells you what like if per like every rows here are the genes. So they will just tell you if this gene is the highly variable ones or not. So they will just predict if there's like highly variables. And then later on, we'll be running the dimension reductions. So basically, they will just I think I also demonstrate this code in like the bulk RNA-seq workshop. So basically, this is just sc dot using the toolkit dot PCA function, and then inside you just put your single cell RNA-seq data metrics here. Then they will just help us like calculate the PCA. They do the dimension reductions of our single cell RNA-seq data sets. And yeah, this is just telling I also not sure how to interpret this, but basically this is just like see how's the PCA to like how's the variance, but I don't think this is important. And then uh this is just a plotting functions of your Scanpy, so they actually won't add or remove anything from of your like end data. So this is just like yeah, included in the um yeah, this is just like included. Like in single RNA-seq, we will be using the PCAs like reduce the dimension and then use like UMAP to visualize them. And then yeah, after we using PCA to reduce the dimension of our transcriptomic profile, we will just like run this code pp.neighbors. So I think this is just like one of the method they use to like identify the clusters. Like I think there's an algorithm to like what's it called the nearest neighbor to like identify the clusters. But yeah, I don't think this is um yeah, I don't think this is you need to know exactly what to do, but you just need to remember to include this. So the final step, I'll be using the toolkit.umap functions. So then they will actually calculate the UMAP metrics for us. And yes. After they calculate, we run the toolkit.umap, we can start plotting the UMAP. So this is a UMAP projections of my single cell RNA-seq data set. And you can just like specify the colors you want. So what I did here, if I specify the colors, because this is a data set that merged two samples. So in my case, how can we identify if there's like batch effect or not? Just see if there's like a cluster that is only like one color specific, and then we will think that's like um there's a batch effect. I think I probably talk about batch effect. It's just like the separations are not based on biology, it's based on the technical part where you do the sequencing. So I would say it's a little bit like this, but I think should be fine. Like to me, maybe this part and maybe this part, but you can still see there's some like orange and blue dots. Like there's some orange dot here, there's some blue dot in the background. So I think it should be fine. So there's no batch effect. Um, so I don't think we need to worry about that. So this is the UMAP part. And then um the next part, when we get the UMAP projections of our single-cell RNA-seq datasets, the later on we need to do the clustering to identify what's the specific clusters. So the we are using they call like Leiden clusterings. I also don't think you need to worry about that. Like what is Leiden clusterings? Um, but we're just using the Leiden functions in the Scanpy's toolkit. So just sc.tl.Leiden. So in this case, they will just help us identify different like clusters here. So if I finish running this, so I think you can look at here, a-data.ops. We would expect they add more information on the .ops layer. So they will have Leiden like 0, 2, 3. So they just like the identified clusters here. So I can use the scampi.plotting functions and then use .umap. And then I want to plot the colors for Leiden. So in this case, I just identify 0 to 23. So it's 24 clusters. Yeah, I think quick review, just like in Python, the index start with 0. So just like remember to like add +1 for number of clusters here. Yeah. So you can plot UMAP over all the columns here. So if it's a numerical columns, they will show a, like kind of like a heat map. And then if it's like this is a little bit like a categorical, so they will show like separate clusters. So just make sure for the clusters arguments here inside the quote, just put the exact column name that you wanted to plot. So it's like pretty straightforward. And yeah, you can even like see how you're gonna do. Like You can also see how you're gonna organize them. So I'm just plotting like to log transform of total counts and then like percentage of mitochondria counts. Um, yeah. So I think I also brought up a concept that in single cell RNA-seq, like this Leiden is a default resolutions of my single cell RNA-seq data set. Just if you look at here. If you increase the resolution, they will increase more single cell RNA-seq data set. So what I will do, I will execute or write a code like this for resolution in like 0 0.2, 0 0.5, 2.0. So for me, I tend to like 0 0.2, maybe 0 0.05, 0 0.0, 0 0.1, 0 0.2, 0 0.3, 0 0.4. I will set more resolution and then I can pick which resolution I want to code the clusters. So this is not arbitrary. So okay, I code like resolution of cluster at different resolutions. So if I want to look at my like the .ops layer, we would expect they give us these numbers of the identified clusters at multiple resolutions. So in my case, let's see. So I wanted to plot the UMAP for all the resolution I called. So if I keep increasing if I keep increasing the resolutions, they will separate more detailed clusters. So I would say, to me, I would like to like pick a clusters around like 13, 15 clusters. So maybe I would go for like 0 0.3 resolutions and then plot like view their gene expression level. But um the tutorials they only pick 0.02 clusters, so they only separate out like four clusters. So I will go for this. But you can also kind of play around because to me, if I wanted to like separate them based on more detailed like sub cell type, maybe I would try to increase the resolutions. And I think I brought this up last week. If I the only downside for increasing clusters are like everything will be messier, and then some PI just hate things complicated. So, but I feel like people should at least know how like yeah, yeah. So yeah. So this is how we can visualize and then select what resolutions I want. So in my presentation, I will always put what is the resolutions, how many clusters I identify, what is the resolutions I use to identify how many clusters. And then I would say, okay, now I identify the clusters. I'm going to use marker genes to map the identity of the clusters. So yeah, this is basically I just picked three resolutions. So a set of marker genes, as I mentioned, these marker genes need to be like well known or need to be like I would say need to be either well known or they're from publications. So these marker genes are from one of the publications. And then in Scanpy, single RNA-seq, if we want to came up with a marker genes, we need to use in a dictionary format. So what is dictionary? It's start with a this bracket and then use column, and then inside the column, just tell them your key value pair, your key value pair for your marker genes. And yeah, so you need to curate this on your own from publications or yeah, I think to me I tend to just like send a screenshot from a publication and then tell ChatGPT to come up with a Python dictionary for a marker gene so I don't need to type this like manually. And yeah, so what like so after I came up with a marker genes dictionary, I curate the list here. I will use the sc.pl.dot plot. And then in this side, just put your marker gene dictionary here. And then for the group by, just tell them which resolution you want to view the expression level. So I pick 0.02 first for now. So if I execute, I didn't execute this code. So they just help us identify specific cell type, what is their signature. So I would say this is lower resolutions. So for example, if I would look at Leiden resolution 0.2, we would expect the y-axis will be longer. But I think the good side for this, we will be able to really separate or isolate out sub cell types. Like um like cluster 0 is like CD4 positive T cells, also have T naive T cells. But like if you look at resolution at 0.2, like the cluster zeros merge all the T cells together. But if I increase the resolution, I'm able to identify like these subset of cells and then also CD8 positive T cells, which, oh, sorry, not this one. Like CD8 T cells are not CD4 positive T cells. But then if we only look at lower resolutions of the cluster 0, we will merge them together. So just like if we increase the resolutions, things will get messier, but we can get more information, more detailed information. You can even do like, oops.
I just had a question.
Yeah.
Um, shouldn't the results for both the dot plots be the same, at least with the top few clusters, because it's the same?
Oh yeah. The clusters, the number of clusters here are the clusters at such resolution. So let me pull this here.
Oh, okay, wait, okay.
So let me put it in the middle. Like this is 0.2, and then this is 0.2 0.20. So like plot so yeah, so the number the clusters here need to match the resolution. So for example, at resolution 0.2, the cluster 0 are this one. 1 is this one. 2 is this one. 3 is this one. But then if we make it if we make it into like if we look at the resolution at 2.0, the cluster 1 now is this one. Cluster 0 is this one. 1 is this one. So the 1 and 0 are actually different locations. And
Right.
then, yeah, in single cell RNA-seq, the UMAP are fixed. So what does that mean? Just what cell type is this are fixed. So 0 are T cells. 0 are T cells at two resolutions. And then 1 is also T cells, according to my UMAP, are also T cells. So, but the one at resolution 0.2 are actually the 0 resolution at the 0 cluster at the resolution 0.02 because they are relative locations. You know what I mean, right? Is it clear? Like the cluster 1 here at resolution 0.2 are actually cluster 0.
Mm-hmm.
Yeah. If you understand this, I think you're better than no, nothing. Yeah. So, yeah. So I would say increasing in resolutions will make us easier, like will give us more detailed informations about what cell type it is. Like if we only look at this one, like resolution at 0.2, we only have four cell types. So then we won't be able to differentiate like I know like cluster 3 are B cells, but we don't differentiate what's the subtype of B cells or maybe plasma cells, for example. They also show their gene signature. And then cluster 0 are T cell, but we can really subset the cells. And then like cluster 1 show multiple like gene signatures here, like CD plus positive monocytes, dendritic cells, or even natural killer cells, and the ILCs. So if we set resolution too low, we won't be able to identify like subclusters. And I think some people also like to use like is that called violin plot? Not violin. I forgot. Is this stack? Why? This one. I think some people like to use let's see. Mm, not this. This one. Stack violin. Some people like to use this, just like the dot plot here, the dots are the fractions of cells that have that gene expression level. So if you want to do like stack violin plot, you can just change the dot plot to like stack violin. And then I think because some people like to visualize them with, oh wait, yeah. So if you do this, they will change it into like stack violin plot. And then it's also the same as dot plot, but violin plot would just shows you like what's the distribution of that cells um that have that expression level. So it's the same, because but to me I feel like stack violin might be a little bit too busy. Like, cause, but it's like, yeah, basically the same, but it just like kind of tells you what's the distribution of the expression of that genes. Yeah. So I would say I prefer dot plot more than stack violin. Yeah. So what is our next step? Like this is my resolution 0.02. So, if you still remember, the format of the AndData.ops is basically just a data frame. So the next part is the tedious part. We wanted to rename the columns of any resolution we want. So I want to rename the Leiden resolution 0.02 because I know like 0 to me looks like it's a T cell. One cluster give me a monocyte vibe, and then two give me a erythroid vibe, and then three give me it looks like a B cell. So basically, what I did here, I basically just renamed the Leiden resolution 0.02 into another new column that I named. So if I wanted to plot UMAP of the cells, we would like what can we change to get a better color? Oh, to like get a better UMAP, we can basically just change the color arguments here into the new named cell type that you found. So then you can just show this and then you can have this here. So then you can came up with this. So I would say if you want to increase the resolutions, you need to like kind of it's just like more tedious. Like um if we wanted to do like resolution, maybe not 0.5. See, if it's 0.5, it's like 19 clusters. I would do 0.2 resolutions. So if you want, what I will do, like you would, if you want to name them as like 0.2 level to get a like a more detailed cell subtype, this is what we can do. Like 0 would be maybe CD4 positive and plus T naive. So yeah, this is a little bit tricky. So sometimes I would do like CD4 positive plus T naive. So yeah, so this is just like you can name anything here. So basically just tells them what is your key value pair. And then one, I think it looks like it's the same. So maybe I will also name them the same way. So in this case, if they're plotting, I think 0 and 1 are these positions. So they will combine them together. And then two, maybe I would name them as, yeah, like this one is a little bit like tricky. So maybe erythroblast. So this is the tricky part because sometimes like if you find out the marker genes like this, um, like here, these genes are not show up in here. So maybe you didn't find the best marker genes, or you can just like make it cleaner. So remove some of the like maybe I will remove this, and then maybe these, I will remove them. I will, if I wanted to like send it to Dr. Louis, I will remove them because I think maybe that will give him too much information that doesn't really help. And I will just remove the list from the dictionary and then replot the dot plot. Like you can just remove a gene. Like you can remove a gene in your list and then replot them. So then you would just like remove some of the genes here. So this is just like what we've learned. And yeah, so this is how we will have to manually identify the clusters here. But wait, two is not erythroblast. Two is maybe I will do okay, I would go pro-erythroblast. And then three, I think I will name them as erythroblast. And then I need to keep doing this. Like so like let's see what's a four? Four, four. Okay, maybe naive CD, and then what's five? Five, I think it's NK cell. What's six? Six also like erythroblast, pro-erythroblast. So you can like you can either make them into the erythroblast clusters, or you can do like pro like slash. But then if you put a slash, they will be separate cluster. They will not merge them together. I mean, yeah, erythroblast. Okay, what's seven? Seven will be plasma cells. What is eight? Eight will be, huh. So you can see like eight have a lot of like different clusters here. You can either like increase the resolutions or just use like monocyte. Just like you can name anything, just like as long as the biology makes sense, or like even the dendritic cell. So maybe that's also a monocyte. So yeah, you can name whatever you want. And then the nine, I think I would go for the same one as three because they got similar like transcriptomic profile, like three and nine. And I also do it for 11, 3, 9, 11. Okay, so now I just need to look at my 10. My 10 look like a B cell. So uh I would just do the 10 is B cell. So in this case, they will create another column at the adata.ops layer. And then if I wanted to give Dr. Louis a more detailed cell type clusters, what I would change is just change into this column here. And then, but I don't like the on data one. I want to make it on the side. So this is how I will hand in with Dr. Louis about the like my cell type. So if you want to like compare like compare with lower resolutions, you can also do that. And then basically, this is just like you can see here, like we identified this position are T cells, and then actually we can separate them based on these two T cells. And then if we look at like monocytes, pro-erythroblast. Like here, this one just kind of like pro-erythroblast, and then it's closer to like monocytes, and then like erythroblast. And I would say I will look back to the gene expressions to look at the pro-erythroblast cluster. It's number two. So yeah, this is the gene expression. So it also like kind of makes sense because it's not a clean cluster here. Like they showed up a lot of different cells, marker genes expression. So I would say maybe this one I won't name it as proerythroblast. Maybe I would name them as like some sort of like unknown. Because I think proerythroblast might confuse Dr. Louis.
This may be a stupid question,
Oh yeah.
but so this is the final result of single cell RNA-seq of 20,000 cells that you gave as a sample.
Yeah. It started with
Okay.
20,000, but now I quality control. So now it's left, it's like like 10K, 11K now, because I
hmm
filtered out 90K. That is low quality.
hmm
So, um, if you change all the threshold up, like in the pre-processing step, your single cell RNA-seq UMAP might look different, because every step will be connected. Like how many cells you leave, how many cells you do PCA, how many cells you keep doing downstream, the UMAP will all look different. So if you like reset the quality threshold, you need to like redo everything here. Just like re-identify, rerun the marker genes, and then manually identify the clusters again. Because like the cluster zero for this number of cells will not be the same clusters for like different number of cells. Yeah. So wait, did I answer your questions?
Yes.
Okay, yeah, okay. So yeah, so I would say this is the basics. I think a lot of the times, I think even in publication, they would just start from here. Stop here if they really don't want to dig that deep into like single cell RNA-seq. They just want to show you, oh, how they identify certain cell types, for example. Because I think in like our project, we want to look at if there's like stem cells. So we will use stem cell marker genes here. Like I will create um progenitor cell. I don't know if I spell it correctly. No, I don't. Or maybe just maybe PC progenitor cells. I will create packs packs 6 and then socks 2. So I will came up with this and I just want to see. Okay, so that just means there's no SOX2 in the gene set. So why? Because if you still remember, I filtered out those cells with like doublet, those genes that had doublet or like too few expression level. So that just means pack SOX2 are not in my gene list, are not in the variable ones. Okay, so if you see here, there's no stem cells here. So yeah, if you don't have like that high of the gene expression, they just won't show up. And yeah. So what did they just show? Like the socks too are not in the A data dot variable layer. The A data are not in the index here, so that's why they show up some error here if you put socks too. So yeah. So I think I would just like this is how we can identify cluster and then use like Leiden clustering. So um yeah, I think yeah, so this is just like how you know. I don't think yeah, I think it's should be pretty straightforward. And then one thing for single cell RNA-seq, the Scanpy, they can also help us identify like differentially expressed genes in each cluster. So for example, I came up with like these clusters. I want to look at what are the top expressed genes in these clusters in the named cell type. So I can just use uh the scanpy.toolkit function and then rank genes group function. So basically they will just identify the differentially expressed genes in each cluster. So you can just run this. They will just calculate it for us. And then you can using the plot function, rank gene groups dot plot, and then also use the same like group that you called. And then this is just like I'm showing the top 5 genes per group. So yeah, I think need to give him some time.
Hey Eric, I have a question.
Yeah,
So how do you know what resolution you want to use? Like typically, how many clusters do you want?
I think to me, I would prefer like 0.2 to 3, like 10 to 13 clusters. Because if like if I can name similar clusters the same name. Like I name cluster 3 and then cluster 9, 11 the same name. So if I'm plotting them, they will just combine into the same clusters. Like they will combine here. If I name them similarly. So if you yeah, so I would say I would pick a cl number of cluster that is kind of like 10 to 15 or to 20. So if they showed similar gene expression level, I would just name them the same. And then on the UMAP, since on the UMAP basically are plotting the colors according to column, this column. So maybe they are different from the light and resolutions, but then if I name them the same way here, they will still be categorized into the same group into the same color here. So yeah, it will be kind of challenge. It's just like more work. For example, if you want to do like, um, if you want to rename like, 0.5, for example, like it's not that challenging. You just need to start mapping them again to like 18, like that, and then. Like to me, you can also like name similar clusters, like the same name. Name the cluster showed similar gene expression of the such cell types marker gene the same name. And then on the UMAP, they'll also combine together. So
So are there any scenarios where you want to use a low resolution?
low resolution, don't really. I think low resolution is just like for like easier to teach. Just like only for like you mean low resolution is this one, right? Or you mean high resolution?
No, yeah, I mean that with like low amount of clusters.
I would say depends on like maybe you just wanted to look at like a broader level of cells, like T cells and B cells. So maybe low resolution is fine. But then if in your case you have different types of cells, like sub cell types, and which they all have like different marker genes, then I think you that's the case you need to like increase the resolutions. Or if you really want to separate something out. For example, at resolution 0. Like resolution 0.5, maybe I still cannot separate out uh like maybe naive T and then CD4 positive T. So but if I really want to separate them out, that's the case when I wanted to increase the resolutions to see if I can really have a like if I do 2.0, I think I also did a 2.01. And then maybe in this case, I will finally have some cluster that only showed up the naive T or only showed up with the CD4 positive Ts. Yeah. So this is when yeah, so if I increase resolution to like 2.0, I think things will just get messier, but then you can get more clear or more defined subcell types. But I think some of the time it's still really hard. And you just need to refine your marker gene list too. If the marker genes are not that specific.
Um,
Yeah.
But want a higher resolution, like completely change your answer for the given data. Like s- like one looks more like it's plasma cells
Yeah.
right now. So isn't that important? Like
One
as
looks like, oh yeah, if you want to look at like where is your one cluster, you need to always like call the UMAP and then see where is that. So the one is here now. So it's different from the one at different resolutions. Because these are just like the identified cluster at such resolutions. And then I think Scanpy, they don't really map like the same resolutions. Like they
okay,
don't really map the ones in, yeah, like yeah, just they don't really map the ones at the same position. Just like the ones at resolution 0.2 is here, but then um like 0.02 is here. And then if you do like other like higher resolution, the one will also be different. So if you want to like um yeah, so if you want to like name the clusters, visualize the cluster, you always need to code them side by side.
right. Sorry, it was a little confusing.
Yeah, is it clear now?
Yep.
Or is it still confusing?
No, I got it.
Yeah, so like the Leiden resolutions here are yeah, I would say these information, like which resolutions like column are all column name, are all like values in a column. So they basically just like, oh wait, this is just like um this is 2.0. So if you wanted to code like for example, so like different resolutions didn't have a line, like not necessarily will have a line like cluster name here. And then the cluster name here will reflect the numbers on your UMAP. So like cluster 2.0, this cells is at cluster 0, this one is at 1. So yeah, so at resolution 2.0, it's like the 0 is this light color here. So yeah, I would say single cell Scampy, they don't really align all the like resolution cluster names. Like this 0 and 1, this number here are all just um the cluster labels that will show on the UMAP, the cluster label that will show on your UMAP when you plot the UMAP function. Yeah. Any other questions? Yeah, I think, um, yeah. So in this case, what I did here, yeah, just go back to the rank genes. I just tell them which group I want to group them. So in this case, I wanted to find out the top regulated genes in my identified cell type at um, resolution 0.02. So basically, I want to look at what are the top regulated genes here among these four clusters. So I run this code, the toolkit function, to get the rank genes, and then I wanted to display like what's the top 5 genes that is highly regulated in my cell type 0.02. So it's just this 4 cell type labels. So these are just they identify the top upregulated genes for like erythroid T cells, monocytes, B cells. So I think a lot of the time, people also think that like this is the first way to identify cluster, like your what is your clusters using like um mapping them with publicly available marker genes for immune cells. This is the first way, and then you manually like check their gene expression level and then name it here. So this is the first way. The second way, some people will also try to rank the genes at these unknown clusters and then look at their up and down regulated, like what is the top up regulated genes, and then see if they can decide which cluster are they. Some people will also do that, but I don't really do that. Just let me show you. Hmm. Yeah. So now if I run this code, I basically just plot a dot plot for the top ranked genes among the clusters identified at resolution 0.2 level. So this is like there's like cluster 0 to 11. So I just wanted to see what are the top upregulated genes in each clusters. So I would say according to this plot, we can see cluster 6, 9, 3, 11. They have similar transcriptomic profile. 6, 9, 3, 11. So they should cluster together. And then like 8 will have their kind of similar clusters, like 8, yeah, 8 is more similar. But then if you look at cluster 0 and 1, they also have similar transcriptomic profile. So 0 and 1 should be named similar, like should be in the same cell type label. So if I look at um so if I look at so what if I want to view what's the color for cell type, like the name cell type, I just switch this out. So yeah, so like according to the this genes, we say like 6, 9, 3, 11 should somehow be the same cluster, 6, 9, 3, 11, and then we also name them similarly. So I would say we name them kind of correct. Like I won't worry about my name, my like identify cluster process. And then 0 and 1 should be named the same because you can see 0 and 1 they showed so similar transcriptomic profile here, 0 and 1. So 0 and 1 is this position on my UMAP. And then so 0 and 1 should be this positions. And then I name them the same. So I won't worry about um that. So I would say these are just like a lot of things we need to like remember how to map the clusters. And it looks complicated, but actually if you like do it twice or third time, it's basically just the same. And yeah. And then for like ranked genes, you can even get the data set out. Uh, you can even take out the like export a list of like DEG lists, just like what we did for pseudo bulk. So maybe you can take this out and then save them into a data frame, and then save the data frame into a new CSV file, and then you can just provide to Dr. Louis, oh, this is your differentially expressed gene list. But if you want to save it to a data frame, don't really include head.5. Yeah. But I would say one bad thing, negative things about like ranked genes group functions is everything is significant because I think adjusted p-value is like really, really high. So I think people tend to just use like top like 100, for example, or top 5. So I would say it's not that accurate as like bulk RNA-seq analysis for DEGs. So yeah. So one thing that's also like pretty nice is we can also plot the colors for certain genes on our UMAP. So what does that mean? They will just plot the expression level of a certain genes we're interested in. So for example, I'm interested in like the top 5 genes that is upregulated at cluster 7. So this is what I do. I just put the genes here, and then also I also wanted to just put a side by side uh like clusters like distributions for resolution 0.5. So then I just wanted to prove that these are the top regulated genes in cluster 7. Hmm, wait, group 7. Wait, this looks kind of off to me. Oh, wait. I think I used resolution 0.2. So this is why I think it looks off to me. Need to be this. Yeah. Yeah, because I'm getting the top rank genes at resolution 0.2. Where did I do? Where did I do? Here. I'm taking the top ranked genes at resolution 0.2. So all the top ranked genes will be at using the resolution 0.2, not 0.5. So did they have any? Oh, wait. Yeah, okay, they didn't yeah. So I'm using the top ranked genes at this column over the resolution, the cluster identified at 0.2. So that's why I think it looks a little off if I do like 0.2. So now if I want to interpret this plot, they're just telling us what are these top rank genes at group 7 show up here. So this is cluster 7, and then now like these genes all show really like upregulated here. So like this is just like kind of look at the top rank genes. What if I have a genes that I'm interested in? So I can just directly use the UMAP functions, and then inside the color just put your gene name. And then you don't need to put that. You can also um change the color of your color map. So yeah, I can just plot CCL5. Or if you're like if in immune cells you wanted to look at like for example COX-1's genes, um so you need to know what's a gene symbol of the gene your interest. There's no gene symbol code. Oh wait, there's also no gene code COX-6. COX-1. There's no gene symbol code COX-1. So how you can know what is COX-1's gene symbol in transcriptomics or in like alignment, you just need to do COX-1 gene symbol. So the COX-1's gene symbol should be also not this one. Um, wait, gene symbol. I think it's something like PT in alignment. Yeah, this is COX-1's gene symbol, PTGS1. So I think a lot of time you need to like be more like specific. PTGS1s are COX-1's gene symbol. So if I want to look at how's the COX-1's like gene symbol, you just need to like get the official name, and you can see there's no clear, maybe at this cluster, COX-1 is slightly upregulated. Maybe. So then this is when I wanted to like make my Vmax kind of have a threshold and then Vmin. So then maybe I will be able to like visualize my clusters more like clearer. No, I don't like this one. Yeah, so like here, if I set the max to only one, I can kind of see here. But then I think in general, COX-1 are not that upregulated. So I think if you want to visualize a gene, you just need to know the actual gene symbol. And then for human, the gene symbol will be all uppercase. For non-human, only the first letter will be uppercase. But then other things need to be lowercase. This is just like how human reference genome did. So if you do like cyclooxygenase gene symbol in alignment in rats, the gene symbol will be this one. Like only the first case is uppercase, and then other thing will be lowercase. And then you can, if you're using a rats or mice, this is when you can visualize certain genes, gene expression level. And I think the final thing is if I make some plots, how I can save this. It's like what we do like for Seaboard. We put the UMAP into a figure variable, and then I use figure.savefig, and then you can just name what's your figure name, your DPI, and then like just add the same thing, and then they will just save a copy for you. And I think, did I link it here? Yeah, I think one thing to remember is if you didn't link your Google Colab, I think it will save it here using to download. But I think I did link it. Yeah, so this now I just save the figure here and then I will just put this into my like slides and then or discuss with Dr. Louis. And yeah, and one thing to note that like because in this case I create so many additional information at my end data. So I don't need to always click it through every time. So I will always wanted to save a copy of my analysis into a new single cell RNA-seq matrix. So what I will do, I will use adata.write, just like df.to_csv, but then I will use the .write functions. And then I will name and like the things I want, just need to make sure to end it with .h5ad. It is just like will save a copy of your pre-processed single-cell RNA-seq datasets into a new H5AD. So then if you next time, if you log back in, you can just directly read this H5AD to get the nData.ops, all the things you already did. So what I do here, the command for read H5AD is here. Scanpy sc.read H5AD, and then the H5AD file name. And then if I do like a data 2. So now my a data 2 will be exactly the same as the a data that I pre-processed them. And I can just always start from here. So I don't need to like run through everything again because I already saved everything. I already saved like everything in here. So if I look at a data 2, if I want to look at the ops layer, just a data 2.ops. This is already the process one, have all the information we want. So if in my lab, I want to save it, I want to share my files to like other people if they want to help me out, I can just share my H580 to them and they can help me start from there, doing more like downstream analysis. But today's lecture is more of a basic ones. And yeah, I think I think I demonstrate, did I demonstrate this? Just if I download a file from Geo, I will just like have a barcode, the feature and metrics. And then what I will do for the read 10x metrics, I will tell them the path where I store these three files and then run read 10x metrics. So if you look at my folder, I save those three files here, the barcode, feature, and the metrics file. And then I just run this code. So now this code, I think they would just like execute. Wait, I really forgot, did I mention this or not? But yeah, let me just do it. And then now this is another brand new data set that I'm downloading from Geo. And then this is uh the Alzheimer's data sets, like a prefrontal cortex, human, single nuclei, but single nuclei basically just the same as single cell RNA-seq. So like in my project, I would just like start walking through the same pre-processing stuff and then make sure all the cells, all the genes are good quality, and then I can use that to identify clusters and then using those cluster to identify like what exactly are that. But one thing to remember is since if you're using a single cell Alzheimer's like brain tissues, the most important thing you need to come up with your own like curated marker genes for brain cells. So that's why like yeah. So that's why like in my one of yeah, in my thesis or like in my like presentations, I will always like kind of tell people what I like um yeah. So just like let me just share. So like as a like a standard like things to report for a single cell RNA-seq data set. So now I feel like you're already pretty familiar with all these steps here, like the metrics, the quality control workflow, and then like for a marker genes, I will always reference to like publicly available marker genes. So if you want to be more comprehensive, you will always need to find out big papers, like papers at good journals, there how they identify their marker genes. And then you will use their marker gene, their curated marker gene to identify the clusters. And then I will always tell them, report what is my Leiden resolutions. And then I will then name them. And then I will identify the cell types. So this is kind of the standard single cell RNA-seq workflow. And then I think just you can try to play around, or if you really find out like some people already did like yeah, like some mice, like you already find out some public data set and you think that can help with your hypothesis generating. I think this is a good method. And then yeah, but then you just always need to keep in mind these are all transcriptomic. So transcriptomic also have their own limitations too. I think it will give you a quick, kind of clear understanding about like how's the transcriptomic level, how's the thing changes. And yeah, any questions? Yeah, okay. If not, I will see you all um next Wednesday. And then I think next Wednesday I will be able to have a car. So I think it will still be in person. And I think I also open up the place to submit your like bulk RNA-seq workshop. And I set a deadline to next Friday. So just like remember to finish this. And for single cell RNA-seq, I didn't have assignment for you. But if you want, you can try to process um the you can try to like play with you can try to play with like how to pre-process like this set of data sets here. I also upload the metric file to the Brightspace. You can just download and then just make sure you specify the path where you put the three files. And then you can try to like click it through. A lot of time it basically just click it through and then um yeah, it shouldn't be hard if you already have a basic notebook here. So I think I import them as a data one. So if you're even lazier, I think I import that metrics as a data one. Yeah, if you're even lazier, you can save a data one into a data. So then you will just overwrite the a data and then you just click it through. And then you can just do the same analysis for a brand new data set. And yeah. And then you can see for this, huh?
Oh, sorry, I just had a question. You can continue.
Oh no, no, no. After you.
So both single cell RNA-seq and bulk RNA-seq have the same end result, right? They basically tell you the gene expression of the cells that you've given for processing.
Yeah, I think the most important thing is we wanted to look at gene expression. But for single cell RNA-seq, we can exactly see how that gene is expressed in what specific cell type. Just like here, we can see how like COX-1 is only upregulated or how like these MZB1 is only upregulated um in the cell types here. And then maybe this MZB1 will be your gene of interest in your project, for example. And then you can see it's like this cluster and then cluster 2470 show the gene of interest, your gene of interest. 2470. So if you look at 2470 at resolution 0.2, it is I think I named this. Oh yeah, I think I just overlapped the A data, but I think it's A data too. So so there's an unknown. But then if you look at if you're interested in these, like so these will be the cells of your interest. For example, if you're interested in oh wait, MZB1, and then you will know like, oh, B cell could be the cell type that you want to focus on. So maybe you will develop your own like cellular work over B cells because you're interested in this gene, for example. So this is how the pros of single cell RNA-seq, other than bulk RNA-seq. Bulk RNA-seq, you won't be able to identify like is this gene specifically upregulated in B cells, or is it in T cells, or is it in monocyte or dendritic cells?
Okay.
Yeah. But you always need to think about the limitations too. Just yeah, yeah. And yeah, and then if you Pia has the funding, you can discuss with him like maybe some parts. If you're really doing transcriptomics, maybe other than like you can do RT-PCR or bulk RNA-seq, but if it's like really a tissue, you can just like suggest maybe, oh, well, we can do some single-cell RNA-seq and then just play with it. Because to me, this is just like click it through. And then a lot of things just like you need to like make your own decisions like this. Where do you want to set like doublet or where you want to filter the cells and then where you want to like filter your mitochondria genes and then yeah and then how's the resolution you want, what are the marker genes you set. So these are just a basic high level concepts and then yeah, I think that's it. Any other questions? And it's not hard. It's like yeah, I learned single cell just like a summer when I was intern. So it's like, and I feel like, yeah, just like you just need to like really know each step, and then you will have a good like yeah. So any other questions? Okay. If not, I will see you all next week, and then um yeah, and then remember to submit the quiz, and then oh, I also need to make the announcement, but um the quiz will be. The next quiz for pandas will be October 29th, right? I think that's what we agreed on. So yeah, just remember to bring just study, but it also won't be hard, so just don't worry about that. Okay. Have a good weekend. Take care.
Thank you.